Hi, welcome everyone to this new episode. I'm here with Mark Rutledge, which is the CEO of World Precision Instruments. Welcome, Mark. Very, very happy to have you on board.
Great to see you, Jeremy, and thank you for having me. Exciting to be here. I've follow of your podcasts, and so it's fun to be here. And I'm always impressed with the guests that you have.
Thank you so much, Mark. Yeah, and I'm very, very happy to have you. Very impressed by your background. So let's maybe start with your background. You have a pretty interesting background on the life science, I would say, industry. So just tell us a little bit more about what you've been doing in the past, and curious to hear about everything.
Sure. Yeah. No, I'm right at about 25 years in both diagnostics and the life sciences space. Most of my career I've spent on the diagnostic side at large companies.
Mm.
Began my career many years ago at GE Healthcare in the radiology, cardiology space, selling, marketing, commercializing imaging equipment. Had an opportunity after 7 or so years to join Roche Diagnostics and kind of grow into executive management roles with Roche Diagnostics, both leading sales and marketing organizations in pretty much all of Roche's business units at the time. So spent some time leading sales and marketing teams in their tissue diagnostics business, their chemistry, immunochemistry business, led their molecular business, and pretty much has spent time in their point of care and all of their divisions. So I had a really good breadth of experiences over 12 years at Roche, running divisions, running commercial teams, and so forth. And most of my journey has been on the commercial side of the business. And it was really only recently where I was able to move into smaller businesses after I left Roche to lead a full P&L where it would be quality, regulatory teams, etc. Spent a few years at a French-based company called Sabia. Sabia is a market leader in protein separation, electrophoresis, diagnostic equipment. Led the Americas and really grew the business nicely in the Americas and South America. And then Jeremy had an opportunity to really lead at a CEO level globally for WPI, and have been here now just over 2 years. It's been a fun journey so far. And what attracted me to come here was really a couple things: the technology potential of the business. And so WPI has a very rich and unique history, and has the potential to broaden the legacy technology and some of the expertise of the technology that we've done in newer growth segments, pharma, CROs, et cetera. So, yeah, really, that's what attracted me to come to WPI. We've got really good runway of growth, and some of the things we're getting to, which we'll talk about in a moment, are exciting.
Well, I'm very. It's great to hear that. I mean, the first question that I have for you is like, I mean, how did you come up to join WPI? Sorry, is that like a company that you created? Is that like a company that you joined? Can you tell us a little bit more about that?
Sure. Yeah. No, really wasn't looking to leave the company I was leading prior to coming here. The WPI has, been around since the late '60s and was founder led. And prior CEO and the prior founder, after the founder passed, the prior CEO had ventured into retirement. And so the private equity firm that owns the business were looking at a new CEO to take the business to kind of the next chapter, and had reached out. And when I did some studying on the business, got to know the private equity firm that owns us today, and really
Mm,
liked how they lead businesses, really liked how they invest in their businesses and drive growth, and how they manage their assets really connected with me. And also, as I indicated, the business has really great potential. And so business has been around for 50 years. Like many small biotechs, they spawned out of a Yale University lab and patented some, early products, amplifiers, and so forth. And, over the period of the next few decades, really the company's construction was what I would call blood and guts physiology, focused on heart, nerve, neuro, epithelia. And in that time, the company had pioneered the first commercial tear apparatus, and also with that brought into the marketplace, in the late 90s, the first micro pump, if you would, to push fluid at really low rates. Um, and over that time, the company added lab supplies and really built a nice little business on tier instrumentation through the Evon product line, a
mm,
fluidics product line, and then what I would call the core or legacy products was lab supplies, surgical tools for animal physiology, etc. When I came on
mm,
board, the opportunity was, and this is another thing that attracted me to the opportunity, was the company needed focus and direction. And so
mm,
one of the first things we did is really set the direction of the company. And that came down to, do we want to be a lab supply, animal physiology, surgical tools company? Or do
mm,
we want to leverage the technical expertise that we have in house, build out our analytical instruments, build out our cell biology instruments in the cell and gene therapy space that pharma is focused on, and really become an innovative cellular analysis, cell biology company, leveraging our great brand recognition in tier measurements. And so that's where we chose to take the company. We started that journey about 2 years ago.
Can you touch on like a little bit like on how I mean how that happened exactly, like when you joined the company? Because and I want to highlight the impact and what is the role of a private equity and what strategy, how they process, like from the time that they decided to join this business, which was at the time led by the founder, and what was the status at the time, and see like what relationship can do you think like the private equity had with that founder, and how did they started to get involved maybe on the strategy, and when did they decided to that it was the right time to have like a new CEO to be hired for that strategy, to execute that strategy, sorry.
Sure. I mean, most private equity firms, have a strategy and a holding time frame of anywhere between 5 to 7 years on average.
Mm,
Can be more than that, of course, or it can be less than that, just depending on how the business value has evolved. Um, but the private equity firm that owns us, the Anderson Group, um, they've held the business for about 7 years now.
wow.
And at the time they were looking for a new CEO, the real need of the business was um, focus and direction and identifying growth opportunities in growth segments, because the animal physiology market, was evolving. It had been evolving. And
Mm,
so when I joined the company, the first order of business was get the business focused on areas of growth. And so the reason I think they sought to recruit me is because I've had experience in turning around small companies. I've also had, with my experience in diagnostics and life sciences, had a good sense for where we could take the technology and how to realize the potential of the technology. And so the private equity partners really, didn't tell us what the strategy was. We went out, did some assessment, and said, here's where we want to take the business. Here's why. And here's the investment we might need either in talent, systems,
Mm,
technology. And here's the return we can get. Most private equity firms, including the Anderson Partners, as long as the return on investment business case makes sense, they'll support it, and we're accountable to pull that through. And so they didn't necessarily dictate the strategy or the focus or
mm,
direction of the business when I joined in January 2022. I was accountable to go identify where we wanted to take the business and why, what investment was needed, and what return we would get, and set up a business case to do that. And we did that, and they got on board. And in the spring, summer of 2022, we started executing on the new strategy, the new direction. And then a part of that was we had to go out and, acquire talent in certain areas of the business that we needed to help me pull through that. So we brought on some new industry experienced executives on my executive staff, beefed up our sales and marketing team to help execute on the commercial strategy,
mm.
brought on a chief scientific officer as well to help build out our scientific strategy. And from there, we've been in the process of moving in the direction of our intentions, and that is to really be a player in early drug discovery and even drug development with our systems.
And for that specific strategy, does the strategy was, I mean, mainly focus on trying to sell existing technology, or try to focus really on the commercial side, or to because there's different type of strategy, obviously, that could apply. Can you tell us about in very high level what's was the strategy?
Yeah. Our strategy was to build upon our cell biology instrumentation and
Mm,
our microfluidics instrumentation while adding some technology to our tier technology, enhancing it, building it out, and bringing
mm,
into market um, upgraded systems. That would mean more automation to those systems, better user interface via the software of those systems, and also position ourselves for the emerging organ on a chip space or engineered model space. And that is
mm,
simply engineering, human organ systems for drug discovery, um, and building out our solutions to support that emerging marketplace. So it was a little bit of both, building out
mm,
existing technology and into newer markets that were
mm.
adopting the principle of tier, and then bringing on some new acquired products that support that via tissue and cell consumables, if you will, um, fluidics, microfluidics pumps for the organ on a chip space. And we're still building our pipeline, our new product development pipeline, as we speak, to continue positioning ourselves to be a key player in early drug discovery, organ on a chip, with to pharmaceuticals, CROs, and academia.
How was the organ on the chip markets at the time that you implemented that strategy? Was it like a how does the competition? How did the competition look like on that market? Was it like a new market just created, or was it like pretty mature? What was the situation?
Very good question. It was very immature,
Okay.
and still is quite um, early in maturity, I would say, even 2 years later, but growing quick. Um, and what was driving the creation of human cells on a chip that could be utilized for drug discovery and therapies was policy. It was and is still evolving as it relates to animal physiology.
Mm,
Cost globally is increasing, as you may know, in utilizing uh, mice and other animals in um, drug discovery and research. And the main driver was, you know, animals can be replicated only so much as it relates to humans. And
mm,
so there are some clear limitations to animal physiology, and the accuracy of drug development pre phase one clinical trial in sole utilization of animal physiologies in research. So getting to accuracy, getting to truly replicating human cells on
mm,
a chip and measuring how those cells react to would be drugs, that was the bigger driver of getting to engineered models. And so technology had evolved to the point where they could get better readouts, better reading of how um, cells react and the efficacy of drugs on how cells react in a true replication of a human organ. And so that
mm,
desire to be more accurate in drug development and discovery was really what was driving, the emergence of organ on a chip space and its growth still today. So, it was it's still very growing because in one dynamic that I think our solutions can bring to the marketplace to help it be can continue to grow is our tier instruments can be used to help with standardization and accuracy of readouts on the cells because today organ on a chip companies that are doing readings and analysis on chip are doing it all differently. Companies are not standardized in the way they are reading the cell reaction on a chip. And so we have the ability, and we're in development of a what we're calling universal tier on a chip, and that will bring standardization to the organ on a chip space. It'll bring automation because it's a very manual process today, and that'll bring consistency, reproducibility of readouts
mm.
from the chip. So
Can,
that'll give you a sense for still how young it is.
can we just like back up like just a little bit just to try to give like a quick overview of the technology itself? Like I would like to better understand like how does that work? We're talking about like the benefits, the type of cells that you can replicate, if there is any limitation, how, if you can just provide like a quick overview of that technology.
Yeah. So essentially, a organ on a chip company like a Emulate or a CN Bio or Synvivo, essentially they're growing cells in culture that replicate, an organ. Um, and so most of the organ on chip companies today have a liver on chip, as they'll call it, or a um, heart on chip or lung on chip, where they actually match or replicate a human system from
Mm,
that organ onto a chip. Um, and that is, a true replication of what the cellular makeup of that human organ is.
mm,
Next phase would be, human on a chip, where they have, replication of cellular makeup on chip, of, human. And so, um, and then they sell and those chips to research organizations or CROs, pharmaceuticals who are testing their would-be drugs in that cellular makeup.
mm,
So the business side of it comes where um, pharma companies, CROs, academic research companies doing, universities doing research on chip will purchase those already made and produced cells on a chip, buy them, and do their research and readouts and assessments on that chip. And so the chip becomes then almost a consumable, if you would, um,
Mm.
to those who are buying it. We come in with our instrumentation to be used as they are utilizing that chip and assessing how the cells react and so forth. Um, does that help a little bit?
Okay. Yeah, absolutely. That's perfect. And so this doesn't replace the lab process, which can go typically like after. It's more like a preventive type of research process that you will start on those chips first to get like maybe more efficient testing, reading information, and allows you to maybe get like a better result because of the fact that you can test like potentially like more drugs faster without like protecting the life of an animal, for example. And then once you identify like those promising drugs, for example, in that specific example, then you will go to that animal testing phase, which is not replaced, but it's more like preventive, right?
Yeah, that's a very good point. And so the FDA has been also clear on this in terms of encouraging engineered models that today not intended to replace animal physiology, more intended to speed up some of the
Mm,
discovery,
mm,
get to um, more cost efficiency in that discovery process, and then almost reflex to animals, if you would, you know? So no time soon will the organ on a chip marketplace replace animal physiology. That will remain for quite some time. It's just a complementary or pre assessment with the ability to do it faster, more
mm.
automated, more cost effectively, before reflexing to an animal where needed.
And how do you guys, I would say, like differ as on the technology itself compared to your competitors on that specific field, for example, unique, I would say.
Yeah. So where we find the value of our instrumentation, our tier instrumentation, um, and the differentiation, number one, for tier and how the electrical readouts are done on barrier tissue, there are not many players in this marketplace, number one.
Mm,
So as it relates to tier measurements, there are a few players. WPI was the pioneer of the first commercially produced tier instrument and has the leadership and brand recognition in all marketplaces that utilize the principle of tier. But even the past couple of years, there's been more players to come into, this marketplace. Axion is one. Applied Biophysics is another that's getting into this space. I think where we compete quite nicely is we have automation in the utilization
mm,
of tier, which the pharmaceutical industry absolutely loves because they can get 96 well plate readout in 3 minutes. And so
mm.
for high throughput, high volume research labs, we are, selling multiple units of our latest launched automated tier instrument because of the speed, because of the throughput, because of the readings they can get off of a 96 well plate. Um, the footprint of our instrument fits really nicely in the incubator as it's used in the lab. That's an attractor. So we have better footprint, I would say, than others getting into the marketplace, as well as reproducible readouts. And so these are some of the areas where we compete quite nicely and are positioned in a differentiated fashion.
Okay, sounds good. Thank you so for sharing that. I think last time we had a quick discussion and you mentioned like some kind of a partnership collaboration with NIH for stem cell quality control, if I recall. Can you just share a couple words about that?
Yeah, so we've in the past year or so, as we have, elevated the value of tier in markets for cell biology.
Mm,
Um, we've partnered in a few areas to broaden and build out the measurement of tier. And one is with we're doing a co-development project with Georgia Tech on bringing impedance spectroscopy to our EVOM portfolio, and that just means you're able to do more multi-frequency sweeps versus one frequency read sweep, which is attractive to higher volume and users. In that partnership, we had an opportunity to broaden it with the NIH as it relates to a stem cell project that the NIH is working on, specifically the Dr. Barty and his division at the NIH. One of the things they're looking to do is bring about a solution to age-related macular degeneration. And so today they're working with pigs on reversing that degeneration and overall blindness. And one of the things they found is that they were able to, with the right stem cells, they were able to inject the stems into the eye of the pig, and they are getting very favorable results as they assess post-injection of those stem cells into the eye of the pig, where the pig is reacting to light, reacting to movement visually. So the next phase is they will try this on humans, and they will be utilizing our tier instrumentation for the quality control of those cells before they inject them into the animal, and at some point here in the near future, into a human eye. And so when we were meeting with the team at the NIH, Dr. Barty said something pretty, impactful to us as we were, scoping the project and the partnership. Is he basically had said, "Hey, in the next 5 to 10 years, there will be a Time magazine article that says we have solved blindness, and here's how we've done it." And he looked at us and he said, "And we're using your instrumentation to quality control those cells to solve blindness." And so that got us pretty fired up and excited about that partnership. And so where we've seen another value of the instrumentation that we're bringing is they can utilize it in quality controlling stem cells, making the cell is right and ready to be injected.
And I mean, how does that work? If you may just expand on that, like are those like real cells, and you use the instrumentation to basically, I mean, plug to those cells to control like the different, different readings and parameters, or are we talking as well like on, synthetic cells that you use like those chip to try to have like a similar type of simulation compared to the real cells? Like, how does that work?
Make sure that so it's assessing the permeability of the cells, making sure the stem cells. So this is different from the cells that are grown on a chip. This is
Okay.
All existing stem cells that are not grown, but they are human stem cells that
Okay.
we are making sure that the right level of permeability in the membrane is ready and will inject into the cells and replace the the blindness of the cells in the eye. And so it's
Okay.
really quite simply just making sure the cell is got the the membrane and the tissue of the cell is ready to be injected. And so
Okay.
that's basically where the value of our instrumentation comes into in the process.
Okay. Can you let's touch on a little bit more like on you mentioned at some point that um the when we're talking about like organ on a chip technology, that I mean there is like we're starting to collect data and you mentioned the fact that from that data you can obviously like automate like the process and getting like even more data. And is there like ways to uh, better read that data, optimize the overall process, like how that data can be used and what kind of benefits can you get from that data, if that makes sense?
Yeah, the software that we have within our tier instrumentation is, has the ability, to not only, to older instrumentation that we had, had a, you know, like almost like a, hard, ejectable, memory device, if you would, with a USB on it. With some of
Okay.
the later technology that we've introduced, we have, an iPad that the researcher uses with our automated EBOM, where they can visually kind of see how the cell is reacting as the system is measuring the electrical readout of the cells. They can store that data, print that data, interact with it, manipulate it in real time with a better, faster, non-manual user interface, which just, as speeds up the process of assessment and quantification, and also makes it more efficient for the researcher in terms of utilizing the data after the readings. So
Mm.
we've tried to, and we've done in the launch of our later version instruments, is improve software to improve the user interface for the researcher, so it is less manual, less cumbersome, and quicker to get readouts with the system. Does that answer your question?
Yeah. Perfect. That's exactly what I was expecting and thinking. Does do you see, like, for example, like, I mean, like right now everyone is talking about AI, for example. Do you see like how the do you see like the AI to potentially help and improve the process? Is that like for the diagnosis of diagnostic of those reading, for example? How what's your thought on
There,
that?
de- there definitely is that potential for AI algorithms to help in the quantification, you know, after those readings or with the data. And this will definitely help with, again, efficiency and productivity. So as manual is a challenge. Overcoming manual systems and processes in any lab environment, has been a challenge. And so what will help continue with automation and productivity enhancing steps in research, specifically, utilizing our analytical instrument or any where AI, could help with predictive readings, predictive
Mm,
measurements to help with drug identification, dosing
mm,
optimization for pre-phase one drugs. And so this is an area we're looking at, quite closely to integrate into our systems as we continue to bring about solutions to improve cost, timing, automation, efficiencies in the drug development
mm,
process. But yes, it's still very, very early, as you know. And
mm.
so I think, diagnostics and life sciences, technology development uh, is kind of, I would say, earlier than other maybe industrial sectors, in adoption of AI and implementing AI into technology. We'll get there, but
Mm.
we're still figuring out how we to bring that in an efficient and cost effective manner. It'll
No,
it'll be
amazing.
it'll be exciting, and even on the diagnostic side, as you've seen a lot of data out there around how AI is already being used on the clinical, on the diagnostic side,
Mm.
to help preventative medicine, preventative diagnosis, as well as to help with accuracy of diagnosis as well. So life sciences will get there, no doubt.
Thank you so much, Mark. Yeah, the last question that I have for you is really like, what's your next steps on your side? Like, how do you see the future? I'm happy to hear that.
Yeah, I mean, for us, we really see the future. Pharmaceutical companies are on a race in developing therapies in uh, neuroscience, so Alzheimer's, Parkinson's, and also cell and gene therapy. And so we really see that there's going to be technology advancements, and we're a part of this as we speak, in driving down the cost of developing drugs, developing therapies, getting to phase one clinical trials faster. As you know,
Mm,
it's very, very costly. It's in the billions, and it's a decade at times for some, pharmaceutical companies to bring a novel therapy to the marketplace. We think with products like tier measurement, we can and with organ on a chip, we can drive down that time, drive down that cost, and
mm,
improve the time to market and FDA approval for drugs getting into the marketplace. And if we can drive down the cost, if we can drive down the time to get there, that just means the right therapies hit those needing patients faster, and then that can make it more cost effective for patients on certain types of therapies. And so that's where we're focused, and I think that's where you'll see the marketplace move, at least in life sciences as it relates to early drug discovery. How can we help pharmaceutical bring the discovery and the development of drugs more cost effectively and faster, more effective, with more predictable end results and outcomes for patients utilizing those therapies?
do you have any, just a follow-up question on this, do you have any estimates on how much faster the use of those technology can shorten like that development cycle? Any specific numbers? I just was wondering.
I mean, we think there's an opportunity for the technology needs to continue to mature for sure. But there's a belief, and this is talked about at many, pharmaceutical conferences here today and over the past year or
Mm,
so, and even into the next couple of years, where, you know, we believe the advancement of the technology that helps on the discovery side, so the pre-phase one clinical trial side, you know, can be cut in half with the
wow.
utilization of engineered models. And so
Wow.
that would be monumental. Now we're
Yeah.
a ways off from that, but that vision is not unattainable with the technology that we're bringing to the pharmaceutical industries and researchers.
Perfect. Thank you so much, Mark. Any last thoughts to share?
No, we appreciate the opportunity to have us and talk a little bit about where we are and where we're headed as a company. Certainly appreciate the time.
Perfect. Thank you so much, Mark, for accepting to be on board today. Thank you so much for sharing like all those stories. Like that was really insightful. Thank you so much.
You bet. Thanks. Let's do it again here in a couple of years and see where we're at.
Exactly. Thanks, Mark.